So I'm Michelle. I'm one of the postdocs in Zhang's lab in the human genetics department here. And today I'll be sharing how we can kind of identify the factors that shape the genetic architecture of complex traits in admixed populations, specifically using simulations. So very broadly speaking, my research interest kind of focuses on genome-wide association studies and GWAS, but I think kind of given the audience, I don't think I'll have to explain what GWAS is in like full details, but just like as a quick reminder, it is a statistical way to identify the loci that are associated with common complex traits and diseases. Yeah. So GWAS have been around for about 2 decades now, and as of February of this year, there was about 7,000 publications, over 780,000 variants and trait associations identified, as well as over 106,000 summary statistics available if we were to look at the GWAS catalog. So kind of one of the major downstream applications of GWAS is that we can use it to build a polygenic risk score or PRS. So given the GWAS summary statistics and individuals' genotype data, we can take the allele dosage at each SNP, weight that by the effect sizes estimated through GWAS, sum it across all SNPs, and then we'll have the PRS for the individuals. So we can use this polygenic risk score to kind of predict an individual's genetic predisposition to diseases like breast cancer, prostate cancer, or like cardiovascular disease. We can use it to stratify the individuals into like different risk categories, or we can even use it to study the genetic architecture across populations. So over the years in the field, GWAS and PRS have been used and studied quite a lot. But there's this major caveat that we need to keep in mind when using the GWAS results, and that is that there's this limited generalizability of GWAS across populations. So this point was brought up back in 2015 by Alicia Martin and others, where they showed that if we look at the prediction accuracy relative to European populations, that this tends to kind of decrease as we move further and further away from the European populations. So this indicates that, the PRS that we use here, it's not going to work well outside of the European ancestry. And they pointed out that this is largely due to the fact that majority of the GWAS being done mostly in the European ancestry cohorts. So it's showing that, whatever results we find through this European kind of European-based GWAS, it's not going to quite transfer well to the non-European populations. So this can be due to like several biological and statistical reasons, but just to point out like 3 of them, the first is that the effect sizes can be different across populations, and it can be more like population specific. And the differences here is it can be due to like, difference in the environment or like the genetic backgrounds of it. And in terms of the genetic background, the linkage disequilibrium, or LD, which basically describes how variants are correlated. This pattern can differ across populations as well, and ultimately, this can affect how well the tag SNPs are actually capturing the true causal variants. The third point is that if we fail to account for like a population stratification properly, then this can definitely inflate the associations and reduce the transferability overall. So all in all, like all of these reasons can tie into creating like this bigger issue of the missing heritability. And in terms of the heritability here, I'm referring to the narrow sense heritability. So where much of the genetic contribution isn't, really captured, especially for the understudied populations. So the issue of the missing heritability problem is that it can limit our, it can limit our biological insight to understanding the mechanisms of diseases or the phenotype we want to study. So then why would kind of populations have these different, like distinct phonetic architecture across populations, even if we're looking at basically the same trait? Like I briefly mentioned, it can be due to the differences in the environment that these populations live in. But part of the reason lies in the fact that these populations experienced a different demographic events recently. So this study published in 2021 by Derbasla and Will Mueller showed that through simulations and by analyzing empirical data, that as the European populations experience this migration out of Africa bottleneck and going through this exponential growth recently, it kind of led them to accumulate this large number of private variants that are only present in the European populations but absent in African populations, and kind of limiting this this number of variants that are shared between the two populations. So this sort of result was observed, not from, just from their simulations, but also in like looking into the empirical data, like the 1,000 genomes, exome data. So yeah, so this work here, they focused on how demographic events like bottleneck and expansion can basically shape the genetic variation in a way that directly affects the complex trait architecture. So kind of basing off of this study, we sought out to see, to see or understand the effects of the admixture on the genetic architecture. So here, admixture is kind of referring to when there are more than one ancestral background within an individual's genome. So if we look at the admixture plot here, just focusing on the Latin American cohorts, we can see that depending on like which part of the continent these individuals are coming from, there's definitely different proportions of the signatures of African, European, East Asian, and Native Americans genome kind of showing up in these Latin American individuals. So the reason why we focus on the effects of the admixture is not because, well, it's part of the reason, but, there's growing number of admixed populations in the world today. But also in such populations, because there are so many, ancestral populations kind of contributing to their genome, it creates this sort of mosaic genomes that's really hard to kind of disentangle and understand the effects of it if we just directly dive into empirical data. So we approached this project or question in two parts where we, did simulations and analyzed the empirical data. But I guess given, the focus of this seminar, which is tools and technology, I figured it would be best to just solely focus on the simulation part of this project and kind of share how we set up the simulations and, how we can, analyze the simulated results to understand the effects of it. So with that, our goal with the simulation is to kind of identify the factors that impact the GWAS performance and accuracy, look at how the genetic architecture changed the heritability, and assess the transferability of GWAS across different populations. Okay, so well, I guess before I get into like the details of our simulation framework, let's first talk about how GWAS is done in practice. So to do GWAS, we first need a study cohort, and once we have like a decent number of individuals who are participating as part of the cohort, then we can genotype their DNA samples, collect phenotypic data on the traits or diseases that we're interested in studying. We'll have to do like some data quality controls. But once we have like a clean set of genotypes and phenotypes in hand, then we can do a statistical test and see, if there are any associations found with the phenotype. So I think doing GWAS in practice is pretty straightforward process. But how will we kind of recreate all of this if we were to do a simulation? Well, one of the advantage of using simulation is that we can pretty much explore a wide range of evolutionary and genetic scenarios, far beyond what's really possible within like the real data. But, you know, if we want to use simulation to understand what's really going on in real human population, we need to set up our simulations so that it captures the genetic architecture of complex traits in real human populations as much as possible. So for that, yeah.
Sorry for asking such a basic question, but I'm not sure exactly how you define genetic architecture. I mean, is that the correlate between the genotype and the phenotype and the disease, or is it something else?
So when I say genetic architecture, it's basically referring to like the allele frequencies, like the effect sizes of the loci contributing to the phenotype. Yeah, or like when we do GWAS, like what is the p-value and that kind of stuff.
Okay,
Yeah, so it's like kind of how the loci or the SNP is kind of what is the shape of the loci, I guess, or the SNP. Yeah, so generally we're just referring to like frequency, basically. Yeah,
I see.
because that can be different across populations, the frequency itself, or like the effect sizes as well.
Okay, I think that helps. Thank you.
Yep. Yeah, so as we want to capture kind of the real human population, we use this well-established Eurasian population demographic model published in 2011 by Simone Gravel. So in their model, they kind of capture key events like how did the affected population size changed over the time, you know, the time of when this migration out of Africa happened, you know, what was the bottleneck looking like, migration between populations, as well as this like exponential growth happening in the Eurasian populations. So using those key parameters from the Gravel's model, we can basically now recreate human genome if we use using simulations and using this specific software called SLIM. So SLIM is a forward in time population genetic simulator that's developed by Philip Messer and his group at Cornell University, I want to say. But what's really great about SLIM is that it allows us to model both the evolutionary processes and complex demographic histories with parameters that we can control. So here I listed like the key parameters that we kind of need to initialize prior to setting up the populations and running the simulations. But basically these parameters it's kind of demonstrated in the SLIM manual, so it's pretty easy to get these parameters. Or you can look at some other, you know, studies or a published paper to kind of play around, you know, to see like what is the mutation rate, or like what kind of recombination rate do I want to set up for your simulation, basically. But of these five, I kind of want to draw your attention to the mutation types parameter, because this is kind of the main reason why we chose to use SLIM to run our simulation. Because there's like other simulation softwares like MS, but that uses like a coalescent, you know, theory to generate the population history. And that doesn't quite allow us to incorporate selection or fitness into the model. Yeah, so looking at the mutation types here, like I said, it allows us to incorporate the selection, meaning that we can model not only how often the mutation occurs in the population, but also you know, the effects, you know, how they affect fitness overall. So for that, we use this distribution of fitness effect or DFE model. And this model is basically kind of following a gamma distribution where it assigns like a range of selective effects to mutations based on their impact on fitness. Okay, so with our parameters, we can then run SLIM for like thousands of generations, and in practice that only takes, I would say, like about an hour or two. Yeah, depending on like how many samples you're trying to create in the simulation. But overall, at the end, we'll have the simulated genotypes of the individuals or haplotypes output as one of the outputs, and that depends on like, what kind of output you prefer to have. Is it like a, VCF file or like a text file, and etc. And because we incorporated that DFE model, we will also have the selection coefficients of the mutations as the output as well. So once we have the simulated genotypes and selection coefficients, the next step that we need in order to do a GWAS is that we need to connect these two to some sort of complex trait or phenotype that we can study, right? So how do we do that? It's not quite straightforward how we would get to the phenotypes with just these two outputs or data. So the first thing that we need to do is we need to assign the effect sizes to the variants. And in order to do that, we will have to use the selection coefficients. And what allows us to connect these two is this Walker model, which is kind of the main model that I want to share with all of you in case you want to, in case you're interested in like doing your own GWAS. So the Walker model is a framework that kind of describes the strength, that links the strength of the selection with the effect sizes of a variant on a trait. So this is kind of the equation of the Walker model. And this was published back in 2010. And just to go over like what each parameter means, the gamma is referring to the direction of the effects. So this can be either -1 or 1. So meaning, so -1, it means that this mutation will have a trait decreasing effect on the phenotype. +1, it means that it has a trait increasing effect. S is basically the selection coefficient, which we have from as one of the outputs from SLIM, so we can just plug that in. So tau here is kind of the key parameter of this model. And this parameter describes the relationship between trait and selection. So tau can range between 0 and 1. And if it's 0, it means that there's no correlation between effect size and fitness whatsoever. And if it's 1, there's a complete correlation, or if another way of saying that is the phenotype you're looking at is the fitness itself. And then we have this random effect drawn from a normal distribution. So I'm more of a visual learner, so it helps me kind of understand the whole framework if I see like some sort of plot. So if we have the trait effect on the y-axis and strength of selection on the x, and if we look at different trait architectures where the relationships between effect size and fitness are varying. So let's first look at tau of 1 and like tau of 0.5. What we see here is that as the strength of selection increases, so does the trait effect of the mutation. And it doesn't matter, what sort of effect does it have? Like, is it a trait decreasing effect or a trait increasing effect? So it's kind of creating this V or U-shaped kind of pattern. But if we look at this tau of 0, there's really no relationship between, so tau of 0 is that there's no relationship between effect size and fitness. And as you can see, the points are pretty much just spread out and not really showing like a clear trend between these two values. So this is the Walker model that, we can use to assign the effect sizes to the variants, just using the selection coefficients from SLIM, and we can try like different values of taus. But in addition to the Walker model, we use this heritability scaling factor. And this factor kind of allows us to control the heritability given a certain mutational target size. So another way of saying is that this ensures that our simulated trait will end up with the desired level of heritability, no matter kind of how many variants are turned out to be like contributing to the phenotype. Okay, so to go over the whole workflow, we first start off with some human demographic model, and in our case, we used the Eurasian population demographic model by by Simone Gravel. We fed that into SLIM so that we can get the genotypes of the individuals. And as we incorporated the DFE model, we'll have the selection coefficients. And using the combination of the Walker model and heritability scaling factor C, we are now able to assign the effect sizes to the mutations. So now that we have genotypes and effect sizes, the true effect sizes of the mutations available, using this formula, we can give a phenotype to the individuals. And finally, now that we have genotypes and phenotypes available, this is pretty much all we need to do GWAS. So using that framework, we use that to investigate the role of admixture in shaping the genetic architecture of complex traits. And because we want to explore, the effects of the admixture, we kind of played around with like different demographic models as well as trait architectures. So here I'm showing all the demographic models that we've looked at, but overall, kind of the big framework of it follows that Gravel's that human demographic model, but we kind of varied the parameters like the time of admixture. So did the admixture happen in distant time, or was it relatively more recent? We also explored different population history. So, did the population just kept expanding until present day? Or was there some sort of, bottleneck that the population went through after they expanded? Or was there some sort of founder effects in the admixed population? And then we also varied the rate of admixture by changing, which ancestral population is contributing more to the admixed population. And the reason why we played around with like these different parameters is depending on like the population history that can affect, how many variants are present in the population, or if it's bottleneck, then that there could have been some sort of selection going on. So yeah, to understand the whole picture of it, that's why we played around with all of these models. So now on the trait architecture side, we explore these three different tau values. So when there's no correlation between effect size and fitness, slight correlation, and then pretty much a stronger correlation for the tau of 0.5. Okay. So now that I've gone over the simulation setup, I've wanted to share like what kind of analyses we can do with this sort of simulated GWAS result. So the first thing that we can look at is a Manhattan plot, and this is what we usually do if we have like a empirical GWAS summary statistics available in hand. And the reason why we look at this Manhattan plot is that it provides us a genome-wide view of the association signals. So, if we look at the donor 1 and donor 2 Manhattan plot, we can see that it's showing this distinct association peaks, by the populations. And this is reflecting, kind of the unique allele frequency and LB structure that exists within the population. But if we look at the admixed population in the bottom here, I would say it's much noisier compared to their ancestral populations. And this is, like I mentioned earlier, this is because, because of their genomes are so mosaic, as a result of the multiple ancestries kind of contributing to their genome. So this what makes it harder to kind of distinguish the true signals from the confounding ones, especially in the admixed population. So this is where our simulations actually come in handy because, we know the ground truth. We can test how ancestry compositions are kind of influencing the association signals here. So if we looked at the overall association patterns, the next thing that we can also analyze is how well can we detect and localize the true causal variants. So for that, we quantified the GWAS power and defined mapping ability. So for the GWAS power, we defined it as the sensitivity, which tells us how effective are we in identifying the true association signal. So, you know, do we have enough signals to detect the kind of the true causal mutation? As for defined mapping ability, we defined it as the precision. So this reflects how precisely we can pinpoint, the true causal variant within the region where the signal was detected. Okay, so as for the GWAS power, if we included all the rare and common variants in our GWAS design, as you can see, the power is very weak. It's very low, and this is because of the weak and noisy signals that the rare variants are kind of contributing here. So if we filter them out and just focus on the variants that have a minor allele frequency greater than or equal to 5%, that generally just improves the power of the detection power right away. So then we also see that the GWAS power changes with these different genetic trait architecture. So when the tau is equal to 0, the overall power is higher than the rest of the tau values. And this reflects that the rare variants here are less likely to be the true causal mutations. So I would say that, a phenotype that sort of follows this sort of trait architecture could be more like a quantitative trait, like height or BMI. But in contrast, if we look at this higher tau value, we see the GWAS power just dropping sharply. So these architectures will kind of resemble phenotypes like more of a disease kind of phenotype like cancer, where the rare variants are more likely to be the true causal variants. Yeah, so because the true causal variants are probably rare under this scenario, therefore we don't have enough power to detect them. So then we looked at the fine mapping ability or precision. And what's really interesting here is that, as you can see, it doesn't really change across these different scenarios. And what it's saying is that, no matter how many variants we find to be significant, so like it passed the GWAS significant threshold, only about 25% of them are going to be the true causal variants. And this is something that we did not know about before. And we were only able to, find this because we used the simulated data where we know the true causal variance. And, with it, we can quantify the performance in ways that aren't really possible if we use the real data. So next, we can use the simulated data to estimate the heritability and assess how much of it is being captured by GWAS. So here I have the cumulative heritability proportion on the y-axis and the allele frequency on the x. Since this is a cumulative proportion, at 100% frequency, it should add up to 1. So the solid line here is showing the ground truth heritability. So for this, we used the true effect sizes and the true causal variance. And for the dashed line here, this is referring to the estimated heritability, where I used the true effect sizes, but only the variance that passed that turned out to be GWAS significant. So whatever gap we see between the solid and dashed line, it will directly tie back to the GWAS power. Yeah. So and as expected, if we look at this tau of 0.5 trait architecture, the gap of between the ground truth and the estimated heritability is largest compared to the tau of 0, where under the scenario we had the very low GWAS power to detect the variance. Yeah.
There's a question from one of the online attendees asking, is the low fine mapping precision only true for admixed populations or also for populations of a single ancestor?
Yeah, so that's a really good question. What was really interesting is that the colors are like indicating the different populations, and it was observed for like all the populations as well. And this sort of pattern, was observed even if we like increase the number of, samples included in the GWAS. So yeah, so like in every cases that we've explored, it was just around that 25%, precision wise. So yeah, so the missing heritability gap is largest under this trait architecture. But also the other thing that I want to point out is that the stronger the tau, the greater the proportion of this heritability can be explained by the rare variants. So looking at the solid line, you can see already the large proportion of the heritability are being explained by these rare variants. But, under tau of 0 architecture, we see that it is mainly the common variants that are contributing to the heritability the most. So, using simulated data, we can measure exactly how much of the trait variance is being explained by the discovered variants and how much of them still remains unexplained. And finally, as for the GWAS transferability, this is kind of what we expected to see, that if we don't match the ancestral or the GWAS population with the target population, it's just going to yield this poor correlation if we look at the phenotype versus the PRS. So for all the results that I shared earlier, it was using this kind of default sized GWAS where we had 5,000 individuals. And there's always this kind of discussion of, what is the decent number of samples or like acceptable number of samples that we need to have if we are doing GWAS. And I think that kind of depends on the phenotype that we are looking at, but I guess a lot of them would say that 5,000 is kind of the acceptable number. But because this is, we're doing simulations, we can just crank that number up to like 100,000 individuals in the GWAS. And kind of what we hope to see from this was that maybe it would kind of, capture or like detect the association, the signals that we couldn't get with the smaller GWAS. But that's really not the case. Yeah, so even with this extremely large GWAS, we are still seeing this poor transferability of GWAS results across populations. So I guess the main message of this would be, just don't transfer the GWAS results. Try to match the GWAS and the target population as much as possible. So yeah, so to wrap up, guess if I were to answer the question of the research question that I proposed, it turns out that the genetic architecture of the admixed populations is very much influenced by the history of the ancestral populations that are contributing, as well as the prevalence of the rare variants. But I didn't really talk about the rare variant side of this project because again, the seminar is about tools and technology. So I basically wanted to share how we could, create a realistic simulation that models the human genetic architecture and trait, where that can very much resemble the empirical GWAS pattern. And we are able to do this using for like a genetic simulator like SLIM, where it allows us to do that realistic modeling by allowing us to kind of incorporate the evolutionary and like demographic processes while knowing the ground truth. So simulations have become like one of the cornerstones in the field of population genetics. And ultimately, I think simulation is needed to kind of bridge the gap between, theory and empirical data, which I believe is kind of like the key step towards developing methods that are, more accurate and more inclusive for the future of the precision medicine. Okay. So with that, I would like to thank my PI, June, our lab members, our collaborator, Arun, as well as our funding source, and all of you for your attention.